Today, I'm going to deal with the prokaryotic DNA replication. I think you already know the basic concepts, so we will try to recap the things that you learned, and let's see is there anything new in that topic. So I would like to share my presentation right now. So the DNA replication, it is the biological process of production of identical DNA helices from a single double-stranded DNA molecule. Each molecule consists of a strand of the original molecule with the newly formed strand. So every DNA which is produced by the replication process will have a new strand which is a parental strand and a strand which is a parental strand and a new strand which is newly formed during the synthesis process of the DNA replication. Prior to replication, the DNA uncoils and the strands separate. A replication fork is formed which serves as the template of the replication. These are basic concepts that you already learned. Now, you may switch off the video so that you can save the bandwidth. You don't have to keep the camera switch on. Here, in case of the replication process or the different steps in the replication, we have the initiation of the DNA replication. Then we have to understand the different structure or structural differences between the different DNA polymerases that is used in DNA replication process. Then the elongations process in the DNA replication, termination, and the overall control of the DNA replication process. These are the outline of the topics that we are going to deal with. So in a prokaryotic cell, you know that we have, especially when we are taking the typical example of an E. coli cell, we have a cell in which the circular prokaryotic DNA is represented here, and in this circular DNA we will have an origin of replication. In E. coli we learned it as ORI-C, and there will be a termination sequence on the other side of the DNA, the circle or the circular DNA. That we will have a termination site in which the DNA actually terminates during the replication process. And from this original cell which contains the circular DNA, during the replication process or when the replication begins, it forms a replisome. Replisome is formed by unwinding of the DNA strands, a localized unwinding of the DNA strands at the origin, and binding of the required DNA synthesis machinery, including the polymerases and different enzymes or the protein factors that is regulating the DNA replication process, which will bind, and a replisome or a unit of replication is formed, which will proceed in both the directions. In case of a circular DNA, we will have a replisome or a replication fork. Two replication forks will move in both sides or in both directions, which will subsequently or finally it will meet at the termination point, and this is the steps that is already described, and the cell elongates as the replication continues. Then finally the two copies of the DNA is actually formed. Then both the concatenase or the closed or interlinked circle of the DNA is removed. Excuse me. Interlinked circle of DNA is separated, or the concatenase of the DNA circle is separated into two separate circles, and then the cell divides into two daughter cells. So this is the overall process of the DNA replication and subsequent cell division, which we know. So in the prokaryotic replication, this is another schematic representation where we have an origin of replication from where the DNA strand starts unwinding and forms a replication fork, and the fork moves on both directions, and finally the concatenate separates, or both the interlinked circles of the newly formed and old one, and that DNA will separate into two separate circular chromosomes. Here we have the origin of replication and the termination sequence, and the same concept of the replisome is actually represented here. Both the circles will, or the replication forks will move in both directions, finally meeting at the termination point, and it forms two complete circles of the DNA. So this is the prokaryotic DNA, basics of the DNA replication. It's the eukaryotic DNA replication. The eukaryotic DNA is actually a long one, or a linear, instead of the circular DNA like that of the prokaryote, it is a linear DNA molecule which contains multiple origin of sites compared to the E. coli where we have only one OREC. Here we have multiple origin of replications, and from that each origin of replication, a replication bubble forms, and this replication bubbles move in both the directions and finally meeting each other, and these strands, this type of localized bubbles or replication forks which forms bubbles are known as replication bubbles, and finally the daughter strands will be separated into two DNA molecules. So this is the replication process in case of eukaryotes, not prokaryotes as the title mentions. So this is another schematic representation which you might have learned in the degree curriculum, where we have a linear DNA molecule, and from the DNA molecule the strand starts its unwinding, and the new strands will be synthesized on the unwound region or the replication fork, in which we will have a leading strand in which the continuous synthesis of the new strand occurs, and the lagging strand on which the discontinuous synthesis of the this is the leading strand in which moves in the 5'3' direction and on the template, and the other one which is on the reverse order where we have the lagging strand. Lagging strand on is the strand on which discontinuous synthesis of the of the DNA is taking place, and that fragments of the DNA which is produced on the lagging strand is known as Okazaki fragments. I think you all already know these concepts. And for both synthesis of the DNA in leading strand and lagging strand, we need an RNA primer which is required to initiate the synthesis of the DNA, because the DNA polymerase is a polymerase which requires a free 3'OH group for extension. So it for starting or initiation of the DNA polymerase or polymerization process of the DNA polymerase starts with an available free 3'OH group, which will be provided by the primase enzyme, which creates a short RNA primer with a free 3'OH group. Without that primer, the DNA polymerase cannot start because it requires a free OH group. And if there is no primer which provides a free OH group, then the DNA polymerase cannot initiate or start the replication process. That is one of the limitations of the DNA polymerase, which this feature of the DNA polymerase will have its own advantages also. During the replication process, we will learn that one. Anyway, this is the feature of the DNA polymerase which is used in replication, and that DNA polymerase always require a free 3'OH group for the initiation of the replication. And when we talk about the DNA polymerases, in prokaryotic systems, we might have learned that we have three different DNA polymerases: polymerase 1, polymerase 2, and polymerase 3, which is coded by pol A, pol B, and pol C genes respectively. So, when we discuss about the DNA polymerase, we have three different types of DNA polymerases in case of prokaryotes, as I mentioned. And that polymerases generally, there are generally speaking, there are different types of polymerases like RNA polymerase and DNA polymerase. The RNA polymerases were used for for the transcription process and other things. And these RNA polymerases, which is also known as DNA dependent RNA polymerases. Usually, these type of polymerases were named as DNA dependent DNA polymerase, DNA dependent RNA polymerase, RNA dependent DNA polymerase, and RNA dependent RNA polymerase. So there are different types of polymerase enzymes which will be using a template, either DNA or RNA. So the dependence is on the template that is used. If it is using a DNA template for the synthesis of another DNA, then it is a DNA dependent DNA polymerase. And the polymerases which is used for replication process in both prokaryotes and eukaryotes is nothing but it is a DNA dependent DNA polymerase, because the DNA polymerases use a template DNA and produces a double-stranded copy of the DNA. Whereas there are DNA dependent RNA polymerases. The typical example is an RNA polymerase which is used for transcription, where a DNA template is used for producing an RNA copy. And there are reverse transcriptase enzymes, you might have learned that one, in which an RNA is used as the template, and from that RNA template, a copy of DNA is produced during the process of reverse transcription. There the template is RNA, so the reverse transcriptase enzyme is nothing but is an RNA dependent DNA polymerase. Then there are replicase enzymes, which is RNA replicase, in which the RNA is used as a template and RNA is produced during the process. So we have different categories of the polymerase enzymes which is used for copying of the DNA and RNA, and they are termed as DNA dependent DNA polymerases, DNA dependent RNA polymerases, RNA dependent DNA polymerases, and RNA dependent RNA polymerases. The DNA dependent DNA polymerases, which is used for the replication process of both prokaryotes and eukaryotes, were discussed during these sessions. And initially, since we are dealing with the prokaryotic DNA replication, we have to learn the details about the DNA polymerases that is used in case of prokaryotes. Here, as I mentioned, the DNA polymerase 1, 2, and 3, which is available in prokaryotic systems, especially E. coli, we have the Pol A, Pol B, and Pol C genes which is coding for that sequence, that particular enzymes. They have different, the subunits were also mentioned. For example, for the polymerase 1, we have a single subunit of which is coded by Pol A, whereas the DNA polymerase 2 is having 7 subunits which is coded by Pol B gene. And in case of the DNA polymerase 3, which is coded by Pol C, there are more than 10 subunits available. This is the molecular weight of the concerned polymerase enzymes, and this is an important parameter when we are discussing the properties of the DNA polymerases. 3' to 5' exonuclease activity, then 5' to 3' exonuclease activity. Exonuclease activity is the ability of a polymerase enzyme to excise off or remove nucleotides from the extreme ends of the DNA, not in between. The strands broken in between is not considered, but if a polymerase is removing the base pairs one by one at the end or from the end portion of the DNA, then they are known as exonucleases. And here 5' to 3' and 3' to 5', both exonuclease activities were present for these both exonuclease activities were present for the DNA polymerase 1, whereas the 5' to 3' exonuclease activity is not present in the other two enzymes, that is polymerase 2 and 3. It is also having some relevance. And we will discuss while we are moving forward, we will discuss why the activity of the exonuclease activity 5' to 3' is not available in these two enzymes. We will discuss it later. But anyway, You have to understand that the proofreading activity of a 3' to 5' exonuclease activity or proofreading activity is present in all the three different DNA polymerase enzymes. Then polymerization rate is another characteristic of the polymerase enzyme. Polymerization rate is the ability of the polymerase to add nucleotides per second. How many number of nucleotides per second is added to the growing strand. So in case of polymerase 1 it is 16 to 20, whereas polymerase 2 it is 40, and the polymerase 3 you may note that it is up to 1000 nucleotides per second. That much rapid polymerization ability is there for DNA polymerase 3, whereas the other two polymerase enzymes 1 and 2 is not having that much efficiency in copying that DNA. Okay, so the next characteristic of the DNA polymerase is the processivity. The processivity of DNA polymerase is the ability of that enzyme to polymerize long stretches of the DNA. For example, the length of the DNA molecule that is produced in a single stretch is much higher in case of polymerase 3, because polymerase 3 is actually intended for specific purpose of DNA polymerase, the entire DNA replication of the chromosome. So starting from one end of the chromosome, it should go on copying till the other end of the DNA. So that means the processivity is the ability of the DNA polymerase to separate or unwind the strand and copy both the strands for a longer duration in terms of time and the length of the DNA molecule that is produced. So the DNA polymerase 3 is having high processivity, means it can go on or keep on polymerizing for a longer period and longer stretches of the DNA, whereas the polymerase 1 and 2 doesn't have that ability. The processivity is not there. It is intended for specifically for some repair works and other things. So the entire copying of the DNA is not intended for polymerase 1 and 2. So that's why the processivity is not there. It will fall off after from the template strand after a short stretch. And that's why these two properties, that is the polymerization rate, high polymerization rate, and processivity of the DNA polymerase 3, that makes the DNA polymerase the major polymerization enzyme in the prokaryotic system. So the other one cannot proceed with the DNA replication of a longer chromosome because the polymerization rate is too slow, and it will also terminate with after copying a very short stretch of the DNA, and it cannot go on or sticking on to the DNA strand for a longer time and copy the entire DNA molecule. So that's the comparison of the DNA polymerases that is used in case of prokaryotic systems. So this is about the main features, and the polymerization subunit only is presented here. The molecular weight is of the polymerase 2, that is polymerization unit only is represented, and the DNA polymerase 2 shares several subunits with the DNA polymerase 3, including beta, gamma, delta prime, and psi. All these units were similar in case of both the enzymes. Now, this is another comparison table in which the molecular weight in kilodalton of the three enzymes is represented. Then the turnover number per minute, how many molecules were copied per minute, is actually represented. The processivity is also mentioned in another slide. Then the number of subunits is again a repetition of the previous ones. And here we have to see that these two activities were already mentioned. 5'3' exonuclease or 5'3' and 3'5' exonuclease activity is actually marked, and the polymerization activity of 5' to 3' polymerization activity is available in all the polymerases. That's why it is a polymerase. So these are the different properties of the DNA polymerase enzymes. Now, there are different domains in the DNA polymerase of the E. coli, DNA polymerase 3 of the E. coli. Just like a palm, comparing with a palm, with the thumb and fingers and or your hand with palm, thumb and fingers, there are different domains for holding the DNA and processing the DNA. So the domain, this domain is the finger domain, this is the thumb domain, this is the palm domain, and this is the template DNA strand, and this is the RNA primer that is added to the template DNA during the copy. So this is a representation. Now let's see the palm domain, or this domain of the DNA polymerase is the main catalytic site which binds magnesium ions and zinc ions, which is required for the binding and processing of the RNA, so the DNA molecules here. So this is the palm domain, which is the main catalytic site which binds the magnesium ions and zinc ions. Then the finger domain is the alpha helical protein, it is the alpha helical protein, and binds incoming nucleotide, then brings incoming DNDP close to the palm domain for catalysis. So this finger domain, this domain, it is having the role of it is having alpha helical proteins, and it binds incoming nucleotides and brings the incoming nucleotide or DNTP close to the palm domain for all where the DNA template is present. So and the DNA template and the DNTPs were brought closer for the catalysis by the finger domain. And the final one, that is the thumb domain, is maintain correct position of the primer. So that is the various roles of these domains of the DNA polymerase 3. So this is another structure in which the DNA B helicase is represented. Then the different subunits of the DNA polymerase 3 is represented here. So here we have alpha, eta, theta, tau, delta, gamma. All the subunits were represented here, and along with the beta clamp or beta protein, beta clamp. So this is the overall structure of the DNA polymerase 3 enzyme with all the relevant subunits. And when we come to the different subunits of the DNA polymerase 3, there is here is the list of the polymerases: alpha, then eta, theta, tau, gamma, delta, delta dash, chi, psi, beta. So these are different subunits. Here these are the molecular weights of the different subunits which is given. Anyway, I'll share the slide to you, so you may go through that later. And this is the different genes which is coding for the different subunits. And this is the important part where the function of each subunit is mentioned. Alpha subunit is which is coded by the Pol C gene. It is having the polymerization activity. Then eta subunit, that is 5'3' proofreading or exonuclease activity, and theta subunit, in this case this is alpha, eta and theta. This is the subunit. So that constitutes the core polymerase. Then the clamp loading protein loading gamma complex that loads beta subunits on a lagging strand at each Okazaki fragments. So these are the tau, gamma, delta, delta dash. This is the clamp loader protein and clamp opener protein. So this is the protein, this one. Beta clamp and clamp loader and opener proteins. This is the region. Now this one is other enzymes, interaction with SSB, then interaction with the gamma and chi, then beta clamp required for optimal processivity. So this is the beta clamp, and this beta clamp is required for increasing the processivity of the DNA, because this clamp will clamp around DNA template strand, and so that just like a ropeway moving on a string, or the DNA polymerase can move on or till the end of the DNA strand without falling off using this clamp, just like a cable car which is moving through a cable rope. So that clamp is created by the beta clamp, and it gives the processivity to the DNA molecule. So this is another representation of the same, and the different subunits were mentioned and their different roles were mentioned, and the initial alpha, eta and theta which constitutes the core proteins, then polymerase 3 and the overall enzyme, the polymerase 3 holoenzyme is having this much, about 10 different protein molecules, that is or subunits, that is used for the entire DNA polymerase, or the structure of the DNA polymerase, and each is having its own functions or roles. So this is about the eukaryotic DNA polymerase, and we are not going into the details, but just to mention that there are 5 different alpha, delta, eta, beta and gamma. These are the different DNA polymerases in eukaryotic systems, and they have, alpha is having the priming of replication of both strands, then delta is used for elongation of the lagging strand, then eta is for that is a elongation of the lagging strand, then beta, that is the DNA repair, the replication of mitochondrial DNA is another one. So there are a lot of other functions that we want to learn in detail, but that will be addressed in eukaryotic DNA replication process. But anyway, we just want to list the different polymerases, or the 5 different DNA polymerases that is used in eukaryotic systems, and our particular focus right now is on prokaryotic DNA replication and the different enzymes that is used in that replication.